All right, so it's a small enough group that feel free to interrupt, you know, put something in the chat. Just speak up. I'd rather answer questions as we go, as you have them, than wait till the end. But I wanted to give you a little bit of background on me. I'm a veterinary pathologist at Virginia Tech. I'm the son of a dairy vet in Ohio, and I went to school at Ohio State. And then I went out to Washington State, where I did a path residency, and my PhD was on the immunology of anaplasma in cattle. So I moved back east here to Virginia 8 years ago. This is a picture of my dad feeding cattle. This Theileria thing isn't super awesome, but I'm happy to be working on it. It's good to be able to talk with folks about cows. So tonight I'm going to talk about Theileria orientalis, give a little intro, and then the Asian longhorn tick, Haemaphysalis longicornis, and a little bit about transmission, what we know about the region, and what's new. So that's just a brief overview of what we're going to talk about. So we talk about Theileria orientalis, but we're mainly talking about the Ikeda genotype. We've known that we've had Theileria orientalis in especially the Southwest in the US, but that really wasn't clinically significant. It's this particular genotype, the Ikeda genotype, that is new to the US. It mainly causes anemia in cattle, and it's of no threat to humans. It's most common along the Pacific Rim, Asia, Australia, New Zealand. Australia estimates they lose $20 million a year annually. And one dairy farm in New Zealand, they estimate they lost $1 million in 1 year during an acute outbreak. We know it's transmitted by the Asian longhorn tick. We've demonstrated that in the US as well. But it can also be transmitted by needles, lice, biting flies, and potentially across the placenta. So any mechanical transmission can transmit it. The tick seems to be the most common way it gets transmitted. It's highly efficient. So the USDA gets pretty excited about Theileria because there are some foreign species, parva and annulata, that causes some pretty significant disease. This isn't that. It doesn't lead to any neoplasia, any tumors. And as I mentioned, there are multiple subtypes. Bufali, we knew we had. Ikeda is the one that we found in Virginia, but in looking further, we've also found a second one, which is Katosi. And in some studies, that one causes disease, and in some it doesn't. We found that one in Virginia, but also Texas, Vermont, and Wisconsin, and in Canada. So for those of you that are familiar with Anaplasma marginale, that's a bacterial disease that's also tick transmitted, and the estimates are about $300 million in losses per year for that one. We think that this could be similar to that economically, but biologically you get acute disease. So when they first get infected, 1 to 8 weeks after they get infected, they get lethargic, they'll have a fever, they'll have anemia, they'll be yellow, icterus, and then they can have swelling along the ventral abdomen. And typically we see about 1 to 5% death loss. There are some herds that are infected and they didn't lose any cattle, so that should probably be 0 to 5% death loss. And there have been some herds where it's been higher than that. Usually those are smaller farms, and 5 out of 20 cattle is higher than 5%. There's also an association with late-term abortions, which I wasn't sure how frequently we saw that, but this fall I have been getting lots of calls from folks about late-term abortions or stillbirths. And we're looking into how that happens, whether that's just anemia from mom, she can't carry the oxygen to the fetus, or if there's something else happening. You know, I don't know about up there, but down here we had some drought conditions, and so grazing wasn't the best over the summer. And some of the veterinarians say that this late term abortion and small calf size can be associated with that drought conditions. So I don't know for sure whether it's all related to Theileria. The other thing I've got bolded here is calf mortality. So calves can die from Theileria orientalis. That's a big problem they have in New Zealand with dairy calves. That's different from anaplasma, where we usually think of up to a year and a half. They're fairly resistant to disease from anaplasma. So we see deaths during periods of stress associated with calving, heat stress, potentially nutritional stress. After the cattle get infected, they get a high level of parasites in the blood. This is anaplasma, but this same figure could be true for Theileria. They get a high level of parasites in the blood, and then the immune response controls it, but it doesn't completely eliminate the infection. And so we have low levels of persistent infection. So once a cow's infected, she's infected for life. But typically the clinical disease, the death or lethargy, those kind of things are during that acute infection, that early infection, and then they just have that persistent infection for life without much consequence. I mentioned, there are herds that they don't even know they have it. So no clinical signs is the most common presentation. Here we've got an example of icterus, that yellow on the vulvar folds there. Icterus, anemia, weakness, abortions, and calf losses. This is a regional vet, Dr. Pat Cummins sent me this picture, and one of the vets has a young daughter and has seen these cows and calls it banana fat. They're just yellow all over, and a bright yellow. Here's another picture of that. And this is what the organisms look like if you were to take the blood from these cattle. They've got these little squiggly lines. Whenever you look under a microscope, it doesn't have the red arrows, so they're a little harder to spot, but that's what we're looking at. So what can we do to diagnose this? One of the things we can do is blood smears and look at it under a microscope, but that can be challenging, and you got to know what you're looking for in order to find it or not find it. So that's not the test that we recommend. We can do serology, so we can take blood and look at the serum for antibodies for anaplasma, but we can't do that for Theileria yet. So what we developed here was a test, PCR test, which many of us are familiar with from COVID discussions, but a PCR test, and we test for both anaplasma and Theileria. The idea is that when you've got a anemic yellow cow, it could be either one of those, and we prefer just to have one test to run rather than running multiple tests. So what can we do about it? Unfortunately, treatment options are fairly limited. Tetracyclines may be effective, but the only time they've been shown to be effective is if you treat early, potentially before you even get clinical signs. Something that is effective is buparvaquone, but that's not going to be available in the US anytime soon, probably never. The reason is there's a really long withholding time, 18 months on the meat and 9 months withhold on calves born to treated cows. So that's just not going to get approval here in the US, most likely. So what we can do is minimize stress. So these animals are teetering on the edge, and one of the things we can do is limit handling. Don't overwork them if you've got a positive animal that's anemic. we're all shooting for a good nutrition plan, but just this is further emphasis for a good plan of nutrition is helpful. And then access to water. Some folks, what they're doing is when they have clinically ill cows, they're pulling them so that they don't have to fight for grain, don't have to fight for food and water to help them get through that initial acute infection. And once they start to recover after about 3 or 4 days, they go back into the herd. So the first time we found it in Virginia, we didn't actually know what we had, but it took us about 6 months to figure it out. So in 2017 in Albemarle County, there were 6 dead cows and the 7th one was sick. They sent the thing to multiple places trying to figure out what blood from that 7th animal to multiple places. And somebody figured out it was Theileria, but they didn't know what it was. And eventually we got the sample and figured out that it was the Ikeda genotype. So this was 2017 in Albemarle County. And then 2018, we found it in Augusta and Pulaski counties as well. And then in 2019, we found it in a bit more. And then last year, we even more. So we're finding it in a few ways. One is the samples that are sent into our diagnostic lab for testing. Another is that we're testing Virginia Department of Corrections herds. And then a third one is surveillance programs. We have one set up with Virginia Department of Ag and Consumer Services where they take convenience samples from sale barns and send them to us, and then we look at how many of those are positive. So this is a rough map. I'm going to show you more maps, but can you guys see that highlight spotlight?
The orange, is that what you're talking about?
No, I was trying to, what about the cursor? I'm trying to point to the orange.
Yeah, we can see the cursor.
All right, so we can see these orange are the Ikeda genotype, and they're running along 81, kind of along higher elevations. The blue squares are anaplasma, and we see those out here southeast, lower elevations, and you can see that the green diamonds are Ketosi, and those seem to be distributed similarly to anaplasma. That even though they're both Theileria, this one seems to be distributed more with anaplasma and less with Ketosi. This is a map that was put together by Alex Telionis, a medical mapper. And these are sale barns in the northern region of Virginia, and the darker red is the higher prevalence in that region. And the top left here is a graph. Overall, the gray line is the number of samples that we got from these sale samples. The green line is the number of Ketosi positive, and we can see there are just a very few, and it's staying the same. Unfortunately, the blue line is Ikeda, and we can see it's going from a very low percentage in the beginning of 2019 to almost a little over 20% positive in October of 2020. And we've got more data, but this is just that cutoff in 2020. So we're seeing that it's there and it's increasing in the number of animals that have it. States that have it, obviously Virginia, West Virginia, I think we're up to 6 or 8 counties that are confirmed there, and it may be more than that. We know it's in Tennessee, and then it's been found in Kansas, which I'll show you when we look at the tick data. That's concerning. We've also gotten samples from Kentucky, Pennsylvania, Maryland, and North Carolina. I'm still working on whether those samples are from animals that got it in that region or they're transplants and just happen to be in that state. Oop, went the wrong way. So red here is where our Ikeda genotype is, and out here in Kansas, and then the blue is where we also have this Ketosi genotype. So just looking at a bigger scale than Virginia. So I mentioned that we were going to talk about the Asian longhorn tick and Ikeda. They're kind of interconnected here. The orange counties are the ones that have both the Asian longhorn tick and Ikeda. Yellow has Ikeda, but not the tick. And the red has the tick, but not Ikeda yet. And so since I've brought up the tick, this is the Asian longhorn tick. These were the first that were found in the state of Virginia. They happen to be on the index farm where we found the first Theileria orientalis Ikeda. They also found these ticks, and the way we identify them is the mouth parts, rather than being long and skinny, they're really broad. There's no color on the back as compared to like the Lone Star tick. And then along the back here, you can see these little ridges, which they call festoons, that are characteristic. And so that's how we know that it's hemophysalis. There's also little extensions off the mouth parts here that's how they got the name longhorn. There's another hemophysalis leporus balestris, which is the rabbit tick, which we've had in the United States for decades, but I think initially we misclassified this tick as the rabbit tick until we figured out that this tick was here. People get pretty excited about it. They write articles like "These savage tick clone armies are sucking cows to death. Experts fear for humans." And I saw that and I immediately thought of my son's comic book, Attack of the Deranged Mutant Killer Monster Snow Goons. That's a little inflammatory the way that's written. It is a potential problem. It has a similar distribution to the Theileria orientalis. It's a little weird in that it reproduces by parthenogenesis, which is a fancy term for asexual reproduction. All of the ticks that have ever been found in the United States of this species are female. There are no males. They don't require a male to reproduce. That's a problem for us because that limits a little bit the control measures, and also one female tick transported to anywhere in the US has the potential to reproduce to be thousands of new ticks without having to find a mate. Typically, the difficulty of finding a mate in low density areas is what slows down the spread of ticks, but this one doesn't have that challenge. Here on the right is a picture from Rutgers, and on the top, this is the larva, nymph, and adult of the deer tick or the black-legged tick. And then the bottom here is the nymph and the adult. They don't have a picture of the larva, which would be similarly sized to the deer tick larva. And here in the center, those are poppy seeds, just to give you a reference on size. Sorry about that. This tick will feed on numerous hosts, but it seems to really like horses and cows. It also, in the suburbs in New York, has been a problem for dogs. It will feed on humans, but doesn't prefer humans, and it'll feed on small rodents and deer. The reason people are really excited about it beyond what it potentially can do for cattle is severe fever and thrombocytopenia virus, which is a virus that we haven't found in the United States, but with a high mortality rate in humans. I think it's improved, but at one point the mortality rate was about 50%. So we really don't want to find that in the US, but that's got a lot of people looking for this tick and looking for that in this tick. And so there's significant national surveillance for the Asian longhorn tick. This is a picture from Dr. Jillian Eastwood. This is one blade of grass, and I count 10 different ticks on there. Some people are talking about getting off their ATV and having hundreds of ticks on them within, a minute or two. And so one of the characteristics is they tend to be in clusters. The scientific term for that is being hyperendemic. But you can see large numbers of them in very small areas. So this is, I guess I'm just going to skip to the current map. The USDA puts together a situation report every month where they list the counties that are positive for Theileria or for the Asian longhorn tick. And this county out here in Arkansas was one of the earlier ones, but here in the last 3 months, we've found a couple counties that are positive in Missouri. And then it hadn't been announced yet, but the 18th state to get it, there's a county that's positive in Georgia now. But the clear winner as to number of counties that are positive for Asian longhorn tick is Virginia. We've been blessed, I guess. Someone took the environmental data, so rainfall, temperature, humidity, elevation, where those ticks survive well in New Zealand and Australia, and applied that to the United States and came up with a map that predicted where we would find the Asian longhorn tick. The different colors represent the different models. The number one, the light brown, is a little more lenient than the number three, which is the dark red. But in all three models, Virginia is going to have the tick pretty well distributed. Everywhere east of the Mississippi is going to be positive. And we can see that, you know, we already know that it's out here in Arkansas and Missouri, and it will get into Kansas. And we know that there's Theileria in Kansas. And so I think pretty much all of the eastern half of the United States is going to have to deal with this, as well as portions of the West Coast. So here's a picture from Dr. Tom Lavelle from Vet Services, VDACs, a picture of the calf ear. And the thing I want to point out is that we've got all three stages of this tick are present in that same thing. And you can see hundreds of ticks. It's fairly common to see those ticks in the ears, and then also in the along the flank and around the udder. We worked with some folks at in Pullman, Washington, USDA branch there, where we were able to show that the tick is capable of transmitting Theileria. Transstadial transmission means that the tick has to pick up the infection as either a larva or a nymph in order to transmit it as a nymph or an adult. It isn't that the adult can pass it to the eggs and then those new ticks can transmit. It has to be picked up in one stage, transmitted in the next. We also injected the organism into cattle and also did tick transmission, and we found that the tick transmission was way faster than the needle transmission. So mechanical transmission may be a cause of transmission, but the tick is really efficient at this. Here I'm just showing that it took 10 weeks for us to get a high level of parasites in the blood whenever we injected it. But whenever we put ticks on cattle, we could start seeing that they were much higher at 3 to 4 weeks. So again, it's faster with tick transmission than it is with needles. This is data from at Virginia Tech, and what this is the number of submissions that we get per month. And we can see that there are two peaks around March and again around October. And so one possible explanation for that is that these are fall and spring calving times. And so, you know, those are periods of stress. And so you're seeing more clinical disease. Another explanation is that we're handling cattle more frequently, so it's easier to get samples at that time. A third explanation, this is phenology. This is when the ticks and what stages of ticks are out and roaming about. And so we know that the larva can't transmit because it has to pick it up as a larva in order to be able to transmit as a nymph or a female. And so the female kind of peaks here in August, which would be just a little bit of time before this peak in clinical cases in September, October, November, and the larva starts to peak here in the spring, a little bit before this peak. And I'd probably say that this year we got more cases in April and May and fewer in March. And so this nymph may explain why we have that spring peak. This is the number of positive cases, and that pretty much mirrors the number of cases that we got submitted. Prevalence at sale barns. We started looking in 2018 in some of the sale barns, and it was pretty low, 1 to 2%, and it's up to 13 in that particular sale barn. A second one was 2%, and then a year later it was 20%. And so we're seeing that we are getting more and more cattle that are Theileria positive. So what do we know? The tick and Theileria are pretty similar distribution. And that fits, that's where the cattle are. And they seem to like the elevation, the tick does, and the Theileria is probably most transmitted by that tick. There are 3 genotypes in Virginia, but Ikeda is the most common. We found it in neighboring states, so it's not just a Virginia problem at this point. One of the things I didn't mention is positive herds have a high prevalence. Well, what we've done is whenever a herd has their first positive, I contact them or the veterinarian to see if I can actually get more samples from that herd to see how many other of the cattle have it. And what we've seen is by the time we get the first clinical case, usually 80 to 100% of the herd is positive. And so the first clinical case, there may be far more that are infected. Some of them will become clinical, and some of them are never going to become clinical. Most of them are never going to become a clinical. We don't have an approved treatment, so it's mainly supportive care. And at this point, the best thing we can do to control it is control of the Asian longhorn tick. Specifics on that, anything that kills most ticks seems to kill the Asian longhorn tick. There doesn't seem to be any resistance associated with the caricides. Some folks are spraying. Usually that's good for about 2 weeks. And so people are putting back scratchers, those kind of things with the caricides at pinch points and trying to do that. The ear tags are great for the ears, but they don't do much about the ticks in the flank. The ticks like high moisture environments. They tend to hang out in tall grasses. And so one of the things you can do to control the tick, if you can afford to give up the forage stockpile, is mow pasture, keep that a little lower, which will dry out the bottom of the plant. And so the tick won't have the high moisture environment to survive. Back to Theileria supportive care during that acute infection, get them through that and you're good to go. And right now, the effects we're seeing of Theileria appear to be libido in bulls during acute infection and a little bit of weight gain in calves during acute infection, but they catch up pretty quick with compensatory growth, maybe slightly lower weaning weights, but not significantly. And the number of abortions that are caused by Theileria, we don't know for sure yet. The challenge with this is once a cow is infected, she's always infected. And so sometimes if you test, she's positive, but does that mean that that's the cause of the clinical disease for her or the cause of abortion, if that's what you're looking at? Usually we like to tie a positive PCR test with anemia to show that, yeah, she truly is clinical. We can also look at we have a semi-quantitative measure, so we can guess, not exact terms, but roughly whether this is an acute or a chronic persistent infection based on the number of parasites that we detect with the PCR. So right now, persistent infection doesn't seem to have any effect on production. It's just the acute portion of the infection.
Dr. Lahmers,
Yeah.
once they show clinical signs and they recover, are they at risk for relapse under other stressful, or are they pretty good from there on out?
So that's a good question. They say there can be recrudescence, but I haven't found an example of that in the US. Having said that, we see the most clinical disease here in the fall, and I can't prove that those calves or cattle all got infected that fall, or that they got infected at some other time. And as they're calving and dealing with that stress and immunocompromise, that they break again. I kind of would need to follow some herds long term, and we're working on that to see whether that we have examples of animals that got through their first that first infection and are persistently infected and then have trouble later. So I don't think so, but we haven't proven that yet.
Okay, thanks.
Thank you for the question. So a question is, do blood titer levels change after they are clinical and recover? Meaning, do they carry a level to impact performance for a lifetime, or does it decrease to a base level? So the number of parasites decreases by like 1,000-fold, and we don't see any performance difference between a persistently infected calf or cow and an uninfected calf or cow. So they go back to a base level that doesn't seem to affect their production after they get through that first infection phase. So what can we do? Tick control, use pyrethroids mainly as treatments. You know, there's some discussion of ivermectin, but if you're using ivermectin heavily to treat ticks, you're going to get resistance and other parasites, and that isn't what we want to do. Especially when the pyrethroids, the sprays, and those kind of things are effective. So for Theileria, we can test for detection to see if it's in the herd, supportive care of anemic animals, and to minimize spread from us, we can try to use single-use needles so that we don't transmit it during vaccination or other times when we're working cattle. So what are we up to? Dr. Laura Hungerford and Alex Telionis are doing some analysis of distribution of Ikeda versus Katosi versus the tick versus anaplasma. And I think we hope to get that data out for everyone sometime maybe November or December. We're continuing to work with VDACs to do surveillance efforts to see if those increase in prevalence continues or if it plateaus at 20%, and also look at the expanding range. We have collaborative agreements with West Virginia, Tennessee, Georgia, and North Carolina, and potentially Kansas, we'll see, and Ohio, to look at other places. Again, it's not just a Virginia problem, and I think it's important that we demonstrate that. We're working to improve diagnostics, add a serologic assay so we can look at antibodies, look at pooled testing so that we can test large numbers of animals more cost effectively and more efficiently. And then working on development of interventions for ticks and for Theileria. Some of those interventions, especially for Theileria, probably aren't going to prevent infection initially, but it's just to prevent that severe clinical disease where we deal with we can potentially lose cows or, you know, have abortions and things that would impact us economically. So I'm just one of the people working on this. I've been working with Charlie Broadus, who's the state vet, and Carrie Bissett, who's in charge of veterinary services at VDACs. Folks here at Virginia, Maryland. Vanessa Oakes is a PhD student who's been working on that, and PMM vets like John Curran and C.R. Gwynn have been really helpful. There have been folks at the USDA here at ARS out in Pullman, and Denise Bonilla and Angela James in APHIS. University of Georgia's collaborated, and many of the regional veterinarians and farmers have been working to help. And we've got some funding from a vet school, One Health grant, and just recently we got some money from a cooperative agreement with USDA APHIS to continue our surveillance. And so, I don't want everybody to think that I'm the only person doing any of this work. I'm just the one that's talking about it today. So that's my quick run through. I'm happy to answer any questions you may have. If you can think of them now. If you can't, I'll gladly answer them later. You can contact me via phone or email.
Dr. Lahmers, what is this going to do to our feeder calf market, to these calves we send out to the Great Plains or Midwest or even Pennsylvania, and our purebred breeders that are selling bulls?
I think that's a good question. So I mentioned this a few times I did a podcast with the American Association of Bovine Practitioners, and we're walking a fine line here between saying, hey, this is important and we need support for this, versus getting people to have a stigma associated with Virginia cattle, because we can't afford to pay a economic price for being in Virginia. So far, I haven't seen that. I've talked with folks in Kansas, and they're not too worried about it, especially because we've demonstrated, at least preliminarily, that there isn't any production effect. There have been some bull tests that were positive, and they actually sold quite well. And as more of the US gets to be positive, having positive animals may not be a negative. If they get past that acute infection, they're good to go. And so worst case scenario is to have a high dollar bull come in to cover a bunch of cattle in an endemic area, and that bull be naive and suddenly get infected and either die. That's the worst case scenario, but they've shown decreased libido. And so you have open cows, or breeding season is spread over a couple months, So having some animals that are already infected may be a choice for areas that know they have Theileria and the tick. They know the cow's going to get infected. So bringing in a positive one actually is beneficial. Another question that I get sometimes is, what about semen? We haven't shown any positive animals, any transmission via semen. I probably need to work with some folks that know they have positive bulls and test semen to see how many are positive. But we haven't seen any evidence here in New Zealand or Australia of AI associated transmission. So for the really high dollar ones that are selling semen, this may not be an issue.
So how long has New Zealand dealt with this?
Australia got it in 2006. New Zealand got it in 2012.
And so it really long-term hasn't been a big negative effect on their production.
No, I mean, it's the first year for these herds that's the big economic challenge, when you may have abortions, you may have loss of brood cows. That's the big economic hit. But as it spreads out, it's, seeing more and more cows. And then if we ever see that calves are significantly impacted here in the US, that in New Zealand is their major economic issue, is the calving loss, calf loss.
I have a question regarding calf loss.
Mm-hmm.
Besides just abortions, we had a number of open animals. Will this Theileria change the uterus, being able to hold those embryos? Are we seeing abortions that, yeah, cattle got bred, but there's cattle that are open now, there's no calf on the ground? Did that abortion take place not at 7 months or 8 months, but did it abort at a month and we just didn't see it?
Yeah, I was just talking with a veterinarian today who was talking about, confirmed bred cows that came back open after 6 months, or 4 months.
Right.
So far we haven't, in conversations with New Zealand and Australia, they haven't seen any long-term reproductive effects. I think that it is purely that acute infection, the cows get anemic and they don't have enough oxygen carrying capacity to support that fetus, but that there's not anything structurally different with uterus, ovaries, anything like that. So they should breed back. Having said that, a year being open doesn't help their reproductive efficiency. But there we got the poll. Excellent. Yeah, so it shouldn't affect them, but we haven't looked at how that's affecting. The other challenge with this one is we're going to see Theileria lots of places, and there have been abortions and sick calves and those kind of things happening way before Theileria ever got here. And so how do we prove what is associated with Theileria, what's directly related to Theileria versus this is just a Theileria positive cow that happened to have an abortion for some other reason? And so, one of the things that we're currently working on is how do we tie Theileria to abortions? What's the mechanism? How's that work? So we can figure out, whether that's what happened to that particular herd or not.
Next question for you.
Mm-hmm.
Thank you for that. Theileria positive cow or cows having been able to get through this and have their calf, that calf coming out Theileria positive or not?
So, most likely not. There have been some studies that said that there was transmission in like 20%, but most say it's less than 1% are positive. So there's two potential explanations, and I'm trying to figure out which is true. One is Theileria doesn't cross the placenta, and if there's an abortion, it's because of anemia, and so all the calves or the vast majority of calves that are born are going to be Theileria negative. The other is that Theileria does cross the placenta, but when it does, it always kills the calf. And so all of them that live are going to be Theileria negative because all of the ones that got infected died in the uterus. So I don't know which is true, but the answer to your question is, yeah, it's you've got better than a 99% chance that calf is Theileria negative. But how long it's negative before it gets exposed to Theileria positive ticks is then the next question.
So it takes about 3 weeks to 4 weeks to mutate once the ticks, if the ticks, cluster of ticks are actually Theileria positive, for them to transfer it to the host animal.
So the tick has to feed for 2 or 3 days. We don't know how long, actually. They haven't done that study yet. But if the tick's allowed to feed, then it's about 2 to 3 weeks after that the animal would be clinically positive. And by that time, the tick has fed and dropped off. And so sometimes folks will say to me, you know, it can't be Theileria because I don't see any ticks on them. It would have been ticks 2 or 3 weeks ago, and they don't have to be on there right now.
So can there be a clinically positive Theileria low animal because there's only a few ticks, and then this animal that has thousands of ticks on her has the potential of being Theileria positive high? Is there different ratings there? Is it overwhelmingly stronger Theileria in a lot of ticks, or is it less positive with lower number of ticks?
So we don't know that answer. What we have seen is that the higher the dose that they're infected with, the faster they get to clinical disease and the lower their anemia goes. And so if they had 5 positive ticks versus 500 positive ticks, I would guess that the cow with 500 positive ticks would be more likely to get to clinical disease and more severe clinical disease. But we don't know what the minimal infective dose is, and we haven't injected them with variable amounts to prove this. There are lots of experiments to be done. We're just trying to figure out how to pay for them and how to prioritize.
So then a animal going to slaughter for meat processing is positive with Theileria headed to slaughter. That's not transferring to human consumption.
Nope, no risk to humans. That's perfectly fine. No difference in anything that we would expect.
Is the Ikeda genotype, does it affect small ruminants?
So it infects small ruminants in that they just this year published a paper where they were able to infect sheep and then feed ticks on sheep and then transmit with those ticks to cattle. So sheep can be a reservoir for it, but they don't get clinical disease. So part of the USDA APHIS money is to test sheep that are in close proximity to cattle to see if we can find positive sheep here in the US. So they may be another reservoir, but you know, cows are close to cows is usually the easier pathway than cows to sheep to cows.
That's good. It's rare that we don't have a problem with sheep, so.
Yeah. It's an uneasy alliance sometimes anyway. So I see in the chat, why do some reports out of New Zealand or Australia show greater performance and economic impacts than what we have seen? So the researcher that does this in New Zealand is Kevin Lawrence, and the one who's doing the majority of the work in Australia is Cheryl Jenkins. And whenever they heard that we got this, they both reached out and we probably talked every couple weeks. What my impression in New Zealand is that they are dealing with mainly seasonal dairies that are out on pasture. And so they are calving at the peak tick transmission time, worst case scenario, and the cattle are out on pasture. And as we know, we push those dairy cattle pretty hard. And so they see pretty significant drops in milk production, those kind of things. And we're dealing with dairy calves, which as a pathologist, I'll say that I think they're different. They're a different animal than a beef calf. They just don't respond. They're not quite as hardy. And so for New Zealand, I think we're seeing that as an issue. The seasonal dairy, on pasture, dairy cows, and high metabolic stress from all that milk production. Whereas Australia, they see it, and you know, they are making that 20 million a year calculation based on 1 or 2 studies, and they studied them short term during infection, but then they didn't follow the compensatory growth where those cattle caught up to their peers. And we're kind of taking the longer view, because ultimately I don't care what happens in 2 weeks. I care what happens whenever I wean those cows, or weaning to finishing. What are they doing as stockers? I don't care on their average daily gain over a 2-week period. I care more longer term. And so in that scenario, we haven't seen the production losses that are described in Australia. So it may be measurement more than actual differences.
Makes sense. Thank you.
Yeah, and I'm in academia, and so we in academia run the risk of being in the ivory tower, where we think purely of science and what questions do I want to answer and how do I get that money. For me, I could push those shorter term losses to demonstrate a higher impact in order to try to get funding, but that would come at a cost. And I'm not sure that the cost to the cattle industry really would, what I was able to gain in money to work on a vaccine may not actually benefit as much as not tanking the market of Virginia cattle. So
I appreciate that. And I think that when it really comes down to it, most producers really want to know what the true effects are going to be anyway.
mhm.
It does not necessarily mean just the short term. They're looking for the long play. So taking that approach is, in my view, the best way to go. So thank you.
So, for us, to me, the impression is that the big issues are death loss and, abortions, stillbirths, open cows, and figuring out how much of this is something else and how much of it is Theileria is going to be a challenge, because we only figure out a cause of about 20 to 30% of abortions. So about 70% of the time, we don't know what it is, but we're seeing them in clusters. And that suggests that it's something more than the one-off abortion. So there's got to be something causing it.
Henry, another question. You mentioned the small ruminants can serve as reservoirs. How about the horses?
So horses get Theileria, but they get Theileria equi, which is a different species. So horses can't do it. But if any of you own horses, this tick can transmit Theileria equi or equine pyroplasmosis. And in the US, the control of that has been, once you diagnose, you either have to euthanize or quarantine the animal. And they got away with quarantine because there wasn't a vector, but now there's a vector that can potentially transmit it. So the equine Theileria may become a significant challenge with this tick as well.
So now that Theileria is here, is there another potential insect vector for it, or just the tick?
Can you ask that again? I missed.
So now that we have Theileria, especially in Virginia, is there another vector that can transmit it, or just that tick and mechanical?
So we don't know for sure. I would love for the folks in Pullman to try all of the ticks that we know we have, dermacentor, amblyoma, those things, exodes, and see if any of them can transmit it. The fact that the distribution of Ikeda and the Asian longhorn tick marry so well make me think that the Asian longhorn tick is the primary one for this genotype. The Katosi is in a completely different area in Virginia, which makes me think that it actually may be transmitted by another tick vector. And in Australia or in New Zealand, they only have this tick. So we know that Katosi and Ikeda can be transmitted by this tick. But that's a puzzle to work out. The short answer is, I think this tick, the Asian longhorn tick, is by far the primary vector for this. It's the main biological vector, but biting flies and lice, other things like that can be mechanical vectors, but they're not very efficient at transmitting. I've had one group of cattle go from 1 positive to 2/3 positive in 2 months over the winter. So it can happen quick.
All right, guys, we have any more questions for Dr. Lahmers?
Got the poll.
Oh, yeah. I accidentally hit that earlier, Jim. I apologize. I was trying to close it and I hit launch.
All right. Let me see. Hang on. I don't know that I can, is it visible? Can you see it?
I can see it. It looks like people have been voting, so.
Okay. Yeah, if you haven't voted, if you go ahead and do that, I can't turn it back on. All I can do is turn it off.
And thank you all for your questions. I appreciate the opportunity to chat with you. And if you have other questions, feel free to get in touch.
So I had one last question here for you before we head out. So we have Theileria positive animals, cattle. That positive animal makes it through the clinical time and comes out the other side, and she's still living and she's now rebred. Can she get more positive if she gets nailed again with more ticks, or she built up immunity to it after a year surviving with Theileria? Is there how long does it take for that immunity, if she is building up immunity, to kick in? Or are we going to see her second calf aborted if she's aborted the first calf, if she gets hit with those ticks once again, or she already built up some immunity to it because she lived through it?
So assuming that she aborted the first time from Theileria, no, I wouldn't expect that she would be likely to abort again, even if she were exposed to more Ikeda positive ticks. She would have to make it through the acute infection. She has built up that immunity and she's constantly getting exposed to more of that because it's circulating in low levels in her bloodstream. And so unless she becomes severely immunocompromised for something beyond just having a calf, I would expect she's good to go.
So that question is leading up to the calling these ones that have aborted, but at the same time, if they live through this time, hey, that open cow could be more valuable to me because she's lived through this, she's built up immunity to it, she rebreeds. Now you have an animal that has lived through this versus other animals that are just now getting it. So we're looking into the issue of calling. Just because she aborted, well, that animal may be more valuable to me if she lives through it a year from now or 6 months from now. I can always breed her for a spring calf if she's a fall calver, or a fall calver could go back to a spring scenario. It's just a judgment call as to, do we get rid of her or do we keep her?
Yeah, I think the rebreeding rate, thinking of it as an ag economist, the rebreeding rate is probably going to be what, if you can get a good rebreed rate and can afford to carry her through that 6 months, she's probably more valuable to her than selling her and trying to replace with something else. And especially if you're happy with the genetics and don't want to turn that over necessarily. Yeah, I think there is something to be said for she has that immunity, and so that's added value for you.
I would hope so. Thank you.
Dr. Lahmers, thank you for taking time out tonight. This was an excellent presentation, exactly what Jim, Corey, and I were hoping for, and it's recorded, and so we'll post this on the webpage